And I have a much shorter career in comparison to the previous speakers. So then there is not many stories to tell. Most of them are done by my postdoc, and then that's what I would like to tell you. So I will share with you a couple of stories. One about cell migration and cytoskeletal regulation, and of course calcium signaling. And the second one is unpublished data that we are drafting, so about to submit at any time. We are a bit delayed by Corona, who is not, and that's what I will show you in the second part. So my main research interest is cell communication and cell migration, and this has been the case since the beginning. And cell communication is a critical phenomenon for life. It's required for many different processes, such as organ development, homeostasis, tissue generation, and the immune response. And that is a particular case because in the immune response, for example, the cell communication plays a key role in determining the outcome of the immune response. In this case, these are just parenchymal cells, and between the signals that they share, there is calcium, for example, of course, AP3, and many other calcium signaling related fun molecules for us. What is striking about the immune cells is that they have to communicate each other, but they also have to migrate large distances, and they do so in very complex environments. And then the coordination of these two processes, these two responses, has to be very tight and fine-tuned. Otherwise, this can lead to diseases. So when either this autocrine or paracrine communication between the immune cells that they need to recognize specifically the molecule to which they are responding to, there is the development, for example, of autoimmunity, or in other cases, a cancer metastasis, when the immune response fails, and then there is a problem in migration, and we ended up with a cancer metastasis. So the combination of these two responses is absolutely relevant. So we are focused in dendritic cells because they play a central role in health and disease. So they can recognize both signals coming from pathogenic pathogens, such as bacterial molecules or viral molecules, and then also they react to danger signals, such as molecules that are released during the death of cells. Then these cells recognize this pathogen-associated molecular pattern or damage-associated molecular pattern. They migrate large distance to the lymph nodes where they activate the T cells, which later on will start the adaptive immune response. And these dendritic cells are tissue resident, and they have to migrate large distances. So, for example, if you have an injury here, so it's a small scar, then you have these dendritic cells will have to migrate a very large distance to the next lymph node to start the immune response. Despite all this, they do this quite efficiently, and that's why we focus on these cells, because they can coordinate their migration in a very efficient way. So, as I said, dendritic cells are already tissue resident, unlike other cells such as neutrophils, which are on the blood, and they have to exit the blood vessel. Dendritic cells are already in the tissue, and they scan the tissue in search for these danger signals. But despite I like very much this scheme by this great scientific illustrator that I suggest you to follow, what happens in the real tissue is much more complex than that. And as you can imagine, analyzing immune cell migration in this context is very hard. That's why a number of years ago, almost a bit more of a decade, a number of laboratories started to generate an approach to mimic certain properties of the tissue and study this response in a more reductionist and simplified way. This is the development of microfabricated tools or microchannels, so then we can mimic the confinement of the cells. That means that the cells are over a roof, so they're sort of smudged what happens in the tissue. We can analyze the speed of migration when they migrate in these straight lines of channels, so then we can quantify the speed of migration. And if we zoom in, we can analyze the intracellular organization of the cells. Even more, we can use them to study cell signaling. For example, this is the calcium signaling in a migrating T cell. And then we can mimic some of the deformation that occurs when the cell is moving within the tissue. So what I will show you now, and this is an anticipated acknowledgement, it was done mostly in the lab of Ana Maria Lennon at the Curie Institute, or the lab of Matthew Piel, working specifically with Pablo Vargas, an independent researcher in his lab at the IPGG Macrophytic building. Then this is the people that I work with, and I have just to acknowledge them, especially Anna, who brought me from Chile to France, and then Mathieu, with whom I learned all the last state-of-art microfluidic techniques. And the catalyzer for this was Pablo Vargas, which, as I said, is an independent researcher in Mathieu's lab. So the part one is about migration. And then what intrigued, the question we had some years ago was, what happens when the cells get activated? So the dendritic cell is patrolling in a resting state, randomly moving in the tissue, and at some point it detects a danger signal. So the question was, what happened with the migration of this cell? And as the great Michael Berridge would say, there is a huge calcium toolkit, and then dendritic cells are not an exception. And then what I will show you here is a couple of proteins that we contributed to this calcium toolkit, which are the mucolupin-1, pannexin-1, and P2X7 receptor. So this is divided in two, as I said, the response to pathogenic signals and to danger signals. So first, we study what happens when the cells were exposed to LPS. LPS is a bacterial molecule, so that mimics the presence of a pathogen. And we will focus on the lysosome, especially Marin Bretou and Pablo Vargas at the time. They were focused on this protein because the processing of the antigen in the dendritic cells occurs in the lysosomes. And as you can see here, the treatment with LPS induced an increase in the migration, the speed of migration of dendritic cells in analyzing microchannels. And when these cells lack the mucolipin-1 channel, now the speed of migration cannot reach the same speed that was in the wild type, suggesting that the lysosomal calcium release is required for the acceleration step. For the sake of time, I won't show you the whole experiments that show, but this is the downstream signaling we unveil. So it's dependent on TFEB, mucolipin-1, then the calcium signaling, and then something happened, and then the speed of migration is increased. What was intriguing is that the lysosomes, here staying in red, in a live dendritic cell, that means that the actin is a GFP. So that we can analyze the dynamics of the actin cytoskeleton in life. You can see that the lysosomes are packed at the cell rear. So then that supposes that the regulation of the response by this mucolipin-1 channel is in a very local manner at the cell rear. So then that led us to think that something was happening at the cell rear. Then we have the danger signal mode. Now the stimulus is ATP, which mimics what happens when the cells break. There is a massive release of ATP. And we analyzed the response in this context, trying to mimic what happens during a trauma situation, which I just described. And we did a 30-minute pulse of ATP, something that, and then watch it out. So then something like the cells would detect the ATP, and then there will be no more extracellular ATP. And we focus on the 2 and the P2X7 receptor, which is an ATP receptor, and a partner protein, which is pannexin-1, non-selective membrane channel that opens up, has been described that is linked, is linked to the activation of the P2X7. So treatment with ATP increased the speed of migration, as you can see here. And when we did the same treatment in pannexin-1 knockout dendritic cells, now you see that the cells are unable to speed up. The resting migration of these cells, as you can see in the two first line, the spontaneous migration without stimuli is similar. So then you have a wild-type dendritic cell and a knockout dendritic cell migrating at a very similar speed, as shown in these first two bars. But then when the cells are treated with ATP, only the wild types are able to accelerate. In between of the experiments that we were doing during this time, we discovered that actually the calcium influx was mediated only by the P2X7 receptor, which was expected. But the pannexin-1 did not contribute directly to the calcium influx. So the question was, what is doing this pannexin-1 channel? Since pannexin-1 has been described to allow the release again of ATP, we thought that the possibility could be that the activation of the P2X7 receptor will lead to the activation of Panexin 1 that will open up and release ATP, generating a sort of autocrine loop that will allow sustained activity and providing the mechanism to accelerate. The prediction was then that if instead of doing an ATP pulse, if that will put a extracellular ATP continuously, then now the cells will be able to speed up. So they're sort of rescuing the defect of acceleration in the Panexin 1 knockout dendritic cells. And that is actually what happens. Now when they treated with the cells with a continuous pulse of ATP, now you see that both cells reach the similar speed, suggesting that ATP treatment induce ATP release to sustain the activity of the cells. So we have two very different signalings. So one mediated by hemoglobin 1, an intracellular store, calcium from intracellular stores, the other one by P2X7 and Panexin 1, so calcium influx from the extracellular milieu. The calcium response when we treated the cells with agonists of these channels, so MLSA1 in the case of hemoglobin 1 or ATP, and in this case for P2X7, the calcium response is obviously very different. And this is what we observe. There is a small peak surrounding the lysosomes in the cytosolic calcium when we analyze the dendritic cells treated with MLSA1, and a biphasic sustained calcium increase when we analyze with ATP. Despite this difference, the treatment with agonists, so in this case MLSA1, increased the speed of migration, and benzoyl ATP, for example, also increases the speed of migration. So then, how these two very different signaling produce the same outcome? And then we didn't think too much, because calcium also regulates the cytoskeleton. So we thought maybe these two responses converged downstream. Despite having very different upstream signals, they converge downstream in the cytoskeleton. So Pablo Vargas developed a technique in which analyzing the migration of the LIFAC expressing dendritic cells, in this case in gray, could be analyzed in the population. So with the help of Mathieu Morin and Paulo Pirobon, they developed a technique in which, after imaging a cell moving within the channels, there was a step of normalization of the size and alignment, generating an average cell. That means that the cell is showing what most of the part of the time where the actin was. In this case, it's cell front. Then we did this in several cells, and then we generate an average mean cell of, which is the average of hundreds of cells, and then generates the behavior of the population. So when we analyze what happens in the immature, non-activated cells, then we see that the actin is mostly accumulated at the front. But when the cells are treated with ATP, there is the appearance of actin at the cell rear. And this was quantified, so the time that actin is spent at the front is reduced when the cells are treated, in this case, with ATP. Inhibition of the calcium signaling and the ATP autocrine loop that I just described prevented this reduction in the time spent at the front, at the back, and then the actin remains at the front, just like in the untreated cells. Suggesting, and well, I don't show you all the data for it, but showing that P2X7 receptor Panexin 1 excess triggers the calcium influx, also an autocrine loop in which Panexin 1 release ATP, that later on activates CAM kinase 2, and then this induces a reorganization of the actin cytoskeleton and increase the cell speed. So, and then the same happened with hemoglobin 1, that I just skipped this data. And then, so we have immature dendritic cells and the resting state, the actin is mostly at the front. And then when treated with either LPS, so the adaptive immune response, pathogen-associated signal, or with ATP, so innate immune response, a danger signal, then the actin goes to the back. So now we sort of unveil that there is the actin reorganization from the front towards the back. It's a conserved mechanism during the mechanism for speeding up of the immune cells, so for fast migration. So let's go to the second part. We are as fast as the cells. Then what happened now during leukocyte polarization? So in Ana Maria's team at the time, there was already, the team was divided in two people working in the dendritic cell migration and also in B cells. However, the cells were not moving in vitro. The they have a lot of similarities when they get activated. So rested dendritic cells will have a certain distribution of the actin and the organelles, but the activation of these cells will cluster the lysosomes at the back. The actin was also relocalized to the back, and the cell now is able to speed up. In the case of B cells, now they are symmetrically organized under resting state, but upon activation, there is the relocalization and the recruitment of signals towards the immune synapse. In this case, you see the clustering of the BCR, the recruitment of lysosomes in blue, and the actin cytoskeleton in red. So Jamie Diaz was studying, a PhD student in Ana Maria's lab, was studying what happens during the early stages of the B cell response. Why? Because the same way that the dendritic cell activation at the early stage is relevant to determine the outcome of the response, the same with B cells. So B cells will detect first the antigen bind into an antigen-presenting cell, then they will, one of the possibilities is to release lysosomes that will degrade this antigen and the cells will uptake it. Then upon processing, these cells will activate T cells that then will get activated and help this B cell back to produce antibodies. So as you can expect, the early stages of this activation also determine the outcome of the response. So Jamie was studying class 1 myosins. So it's an actin-based molecular motor that links the actin cytoskeleton to the plasma membrane. That means that the recruitment of actin towards the synapse could be dependent of these myosins due to the role of this myosin linking the actin cytoskeleton to the plasma membrane. So then that's what Jamie did. That's what the general framework of Jamie's question. So here we use again another reductionist model. We replace the antigen presenting site by latex beads, and then when the latex beads is coated with the antigen, you can see that the B cell extend protrusions and covers up this bead. So now if we downregulate the expression of one of the proteins of the myosin 1, the prediction is that the protrusion should be smaller, because now the actin is not able to bind the cortex, and therefore the cell is not able to do this protrusion. And that is exactly what happens when there is the knockdown of myosin 1C, one of the myosins. Now you see that the membrane is smoother and that the protrusions are smaller. But in a very unexpected manner, when she analyzed and analyzed the MYO1E knockdown cells, now the protrusions were bigger. And that was a big surprise, because we didn't expect this. We were expecting that if we remove this protein, now the protrusions are not able to form because the actin is not able to link to the plasma membrane. Therefore, the protrusion should be smaller, which is the case in myosin 1C. But the myosin 1E were bigger. So we didn't think too much again, because the difference is quite striking. One is called a short tail myosin, myosin 1C, and the other one is a long tail, because it contains these extra domains that allow protein-protein interaction. So the question was, who is the molecular interactor, of which is the signaling under this protein that is different? So we we did a screening for molecules that interact with these ones, with these proteins, and we find out some hits only for myosin 1E. So with the help of Stephanie Miserei, we validated these hits, specifically one, which is PLC gamma 2. PLC gamma 2 is a key protein that is activated downstream of the BCR receptor. So upon engagement of the antigen, the BCR receptor undergoes some conformational changes, and then triggers the downstream signaling that includes PLC gamma 2 phosphorylation. So with the help of Stephanie Miserei, we did just to hybrid assay, and she showed that the myosin 1C does not interact with PLC gamma 2, but myosin 1E does interact with this protein. And I validated this by co-immunoprecipitation, and then you can see here the immunoprecipitation of PLC gamma 2 pulls down myosin 1E. So then what's happening? That's the question. What's happening now? So we know now that myosin 1E interacts directly with PLC gamma 2, but now what is the outcome of this response? We know that the PLC gamma 2 produce IP3 and DAG, and then the IP3 will lead to the increase in the cytosolic calcium. So we analyzed easily as a readout of the activity of PLC gamma 2, we analyzed the calcium, cytosolic calcium. So when the antigen is not bind by any, the PCR is not bind by any antigen, the cells that are in contact with this bead, they have very few oscillations. When now the beads are coated with the antigen, now the cells have much more oscillations. So then there is the trigger of a response. And then when we inhibit PLC with this pharmacological inhibitor, then there is no response at all. And then by the end, you will see that ionomycin shows that cells are properly loaded. So we show here that the calcium signaling were dependent on the antigen, and then this process was dependent on PLC gamma 2. Now, what happened with the myosins? Here you have again a cell responding towards a bead that contains the antigen, so you see several oscillations. Now, when we knock down the myosin 1C, the one that has a lower response in the protrusions, we also see a reduced response in the calcium. So the cell is not able to sort of sustain the activity in this part, especially. Then, if we look at the myosin 1E knockdown cells, now we see that the cells have more oscillations, and actually they are bigger. And then at the beginning of the response, the frequency is very, very high. So then suggesting that myosin 1E actually is inhibiting PLC gamma 2. So that when we remove myosin 1E, the response is increased. So we analyzed in deep the calcium signaling. So we generated these sort of profiles in which we smooth the activity of the cells or the frequency of them, which allow us to pool several cells that they have, of course, very different behaviors. And what we observe is that myosin 1E knockdown cells, they have an increased number of oscillations, so the frequency is increased, especially at the beginning of the response. The cells are hyper-responsive in everything that we have tested by now. I'm not showing you what happens with the lysosomal polarization or with the actin polarization, but all these phenomena are increased. So the prediction is that if there is more calcium, because probably there was more IP3, then also there should be more deacylglycerol. And so we use a molecular probe. So this is a PKC delta domain that binds specifically DAG, and then you can see that the acylglycerol production is limited to the synapse, so where the bead is, that you can see here, the white circle. And then we analyze what happened with the MYO1E knockdown cells, and again, we observed that the response was increased, as expected. Where does this calcium comes from? Where? Then first we tried the inhibition of PLC in both cells. So I already showed you in the SI control, but here is also for the myosin 1E knockdown cells. And then you can see that the inhibition of PLC completely abolished the response. Where is the calcium coming from? There is an intracellular storage component and also influx. As you can see here, the chelation with the extracellular VAPTA reduced the response to only the beginning, probably because the cells are unable to refill the intracellular storage. So we have that the activation of the PCR receptor with the antigen induces the activation of PLC gamma 2. Something happens with the myosin 1E, and then when this myosin 1E is not there, we have more calcium, probably as a consequence of more IP3 and more deacylglycerol. So then we wanted to analyze better what happened at the synapse. What is happening with the PLC gamma 2? Maybe it's not just that there is a change in the phosphorylation, but maybe a change in the intracellular distribution of the PLC. So we changed the model from the beads, and then now we coat the antigen on the surface of a glass. So now the cell will recognize the glass as the antigen, and then they will generate these beautiful spreading cells that with a radial synapse, the classical typical synapse with a central SMAC and a peripheral SMAC. The lysosomes are polarized to the center of the synapse in the case of the wild type or SI control cells. The myosin 1C knockdown cells, they have lack of polarization, the cells are not properly spread, and the lysosomes are not properly polarized. And the myosin 1E knockdown cells, they spread more, and they have increased and accelerated polarization of the lysosome as expected. What happens with the calcium? Here you see that the cells, when they are in contact with a surface that only contains an adhesion molecule, The cells practically in the left, they don't oscillate. And now when the surface is coated with the antigen, they do exhibit oscillations in the calcium signaling, and the calcium, cytosolic calcium, and then quickly spread upon contact with the surface, as these two cells that arrive show. So now, what happens with PLC gamma 2? So we transfected these B cells with PLC gamma 2 GFP construct and analyze what happened at the synapse. So to do so, we use TIRF, which allows TIRF microscopy, which allows the analysis only of the surface, so the membrane contact the glass. Here we can see the appearance of these small black dots after the image processing that are called PLC foci. So these foci are transiently appearing in the what will be the synapse, and they are very dynamic. And so there are some hot spots where they appear more often, but they don't stay for long. As you could expect, when the myosin 1E is knocked down, now we see more foci, very hard to quantify anyways, and they stay longer. So this is a sort of profile in which you can we can transform this radial cell in only one line. So this is the center of the cell, this is the border of the cell, and then you can see that there are more dots and more darker. So they are darker because they spend longer time in the case of the myosin 1E knockdown. In order to have a better analysis, we repeated the same strategy, but now in fixed samples, just to see where the PLC gamma 2 was, and then we found that indeed the myosin 1E knockdown cells, they have increased number of PLC foci per cell area. This suggesting that the myosin 1E is sort of hijacking the PLC gamma 2 from the synapse, and when we remove the myosin 1E, then PLC gamma 2 is more prone to arrive to the synapse and can stay there. We analyzed where was the myosin 1E during the response, and actually it reaches the synapse as shown in this image. So then this is a transfect cell, but fixed afterward with the myosin 1E ERFP tag protein. Okay, so if myosin 1E is downregulating the activity of PLC gamma 2, then we should see the same in wild-type cells just transfected with myosin 1. And then you can see here that the cell in the left, is highly expressing myosin 1E. The one on the right expressed at an intermediate level. And the one in the middle here is not expressing at all myosin 1. And the oscillations in the calcium are expected. So then we have more oscillations in the green cell that correspond to the non-transfected cell here in the middle. Then we have a bit more of oscillations in the cell that express low levels of myosin 1E, and almost none in the cell that express high levels of myosin 1E. But this experiment can be interpreted in different ways. So we went further, analyzing what happens with the myosin 1E, and we cut the tail of the myosin 1E. If the tail is the place where the interaction occurs, then if we cut the tail, there should be no interaction and therefore no effect. So now we repeated the experiment, and we observe again that the cells have increased oscillations in the cytosolic calcium when they touch the surface. And so here the myosin 1E knockdown cells, they have more oscillations, and the magnitude of the oscillations are bigger in comparison to the control. Now we cut the tail of the myosin, and now it contains only the motor domain and the neck. As you can see now, it's the distribution that normally is in the cortex now is only cytoplasmatic, suggesting that the myosin is not able to reach the cortex, which is exactly what we want. And then you can see that the transfection of the cells with this myosin 1E delta tail mutant almost does not affect the calcium oscillations. On the other hand, when we now transfect the full-length myosin that goes to the cortex, as you can see here, now we do see a significant reduction of the oscillations in both cases, in the site control and the myosin 1E knockdown cells, really showing that the full-length myosin is the one that, via this protein-protein interaction domain, prevents the activation of PL sigma 2. So as a summary of this second part, then we have shown that the myosin 1E plays a negative role during B cell activation, specifically interacts with PL sigma 2 and inhibits its function in B cells. The lack of this myosin that probably, as I said, is hijacking PLC from the synapse and put it out. Then if we remove this myosin 1E, now PLC can stay longer and have a sustained activity at the synapse, and therefore there is an increased calcium signaling and also deacylglycerol, which subsequently leads to an increased cellular polarization of the lysosomes, the actin, and the other organelles that I analyzed, but I didn't show for the sake of time. So what happened, so in a bit of perspective, some of the things that I observed and that are part of my research interest is what happens with organelles. So in this very fast acquisition, you can see what happens with the cytoplasm and the nucleus. And then in this case, at least they are perfectly coordinated. However, there are other organelles that are that the calcium doesn't increase at the same time at the cytoplasm, and that is the case for the ER and the mitochondria. Another thing that I observed with this second scale analysis, we observed that the cell is spreading, as you can see in this chymograph, and then the cell is, this is the same cell, and you can see the increases in the calcium, so some oscillations. But now when I analyze with a faster acquisition rate, so now to the millisecond level, I hope you can see, but where the arrow is, there is a small calcium increase. You can see it's very local. There you go. And then this is another movie with a zoom in all in that region, so you can better observe what it is. We don't know yet what it is. If it is a microdomain, a nanodomain, a wave, we don't know, but it's very interesting to know, to study what happens with this local increase. I'm very keen to study what happens with the local calcium. And actually, this has been studied already here where I am now, in the UKE, by the group of Andreas Buse, specifically by Bjorn Dirks. So then you see this T cell now, when then at some point the bits arrive, and then you will see an increase in the microdomains. I hope it's working well. It doesn't look very nice. Something happened with the video. Anyways, you will see the increase of the calcium at the synapse. Apparently it's not working well, but the paper is published, you can check it down there. So more migration. So now I moved from Paris to Hamburg, and I just arrived. So everything was, of course, delayed because of Corona. But now finally I am in Hamburg. It's a very nice city. I'm at the UKE, at the UKE Universität Klinikum Hamburg-Eppendorf Medical Center, and specifically located in this building in the IBMZ, in the Institute for Biochemistry and Molecular Cell Biology. And I'm starting now my own team, so I'm looking forward to meet my recently started team. So everything is new and everything is starting from scratch, but we are very enthusiastic to do so. So I want to formally welcome Lisa and Johan to my team. And my team will be dedicated to study, again, cell communication and migration, which, as you can see, is very, what I really like. And so if you are a passionate and collaborative researcher, or if you want to become one and you want to join us, please just drop me a message. So what will we study? Again, this model that I really like, then you can see what we have already published is what happens when the cells just detect the antigen or the danger signal, in this case, LPAs or ATP. Then they speed up, they undergo fast migration. But then also they undergo an exquisitely intricate molecular program that induce their maturation, and they change the expression of receptors, and now they are able to migrate to the lymphatic vessels upon detection of the chemokine that is in the microenvironment. So in the lab of Mathieu, we have a study in the multi team led by Pablo Vargas, what happens, how the cells coordinate following the chemokine gradient and the obstacles of the microenvironment. This is we have studied for the last years, and I would like to study what happens also, because the cells do not migrate alone, but they have to communicate each other and with the environment, and that's what I will like to study and develop in my team. So we developed in the last years a new macrophytic chamber that allows the generation of gradients, as you can see here. And then we can analyze the trajectory of hundreds of cells following this chemotactic gradient. And what has not been done, because it's not easy, of course, is the analysis of the calcium signaling during this process. So this is something I started to do, and I will continue to do here. So you can see here the changes in the cytosolic calcium in this migrating cell towards the gradient. I will kind of make a mix of all the expertise I brought. So then during my short career, and then I am very interested in organelle crosstalk, calcium signaling, trying to test some old friends, nice hypotheses for the organelle crosstalk, and especially with my recently discovered love for the ER. So then it's a very interesting organelle. It's the main source of calcium in the cell, and that will be focus of my research. So in a complicated year, we were already discussing with some friends about how to improve conferences. And then now we will have one virtual conference too for the GECS, which I'm very happy for. So the GECS is the Junior European Calcium Society Board, and that is integrated by a bunch of very nice guys that you can see here. And then, but we have to acknowledge that the one that started this was Jan Paris many years ago when we met in the Sunday River in the Gordon Conference, extinct Gordon Conference in calcium signaling. And he said that maybe more young people should join and so on and so forth. And finally, we have a board and we are doing a lot of activities. We have two spokespersons, Bjorn and Maleni, but other people that are very involved and the things are running very, very nicely with it. So we are organizing. I'm still member of the board, but now since I am appointed as a group leader, now have to drop the GECS. So we are organizing this meeting. That will be my last activity as a member of the board, and there will be some opening seats. So then if you would like to join, please reach us and then communicate to us in the Slack. And we'll be very happy if you join the meeting. We are preparing a meeting that will be for sure gender balance and as diverse as possible, and it's prepared for early career researchers, which is the main idea of the GECS. Finally, what I wanted to just briefly share with you is that, as I said, with a bunch of friends, we were already discussing how to evaluate, how to improve conferences, and one of these possibilities was to go virtual, because then allows people to have, I don't know, is better access for people that cannot fly for lack of visas and many things that we are generally not aware. And then when nobody thought this could be possible, Corona came, and now well, you see that this is pretty much possible. So I would encourage you to give us as many feedback as you can in this preprint that is published. And with that, I would like to acknowledge you for staying here, for watching this, and for the organizers that are doing a great job organizing this seminar.
All right, thank you very much for the very nice imaging and presentation. So now move on to the question session. The first question from Martin, given that they use different signaling system, is there additivity if LPS and ATP are applied together?
That is a great question by Martin. So yeah, actually we tried putting both at the same time, and there is no additivity. So the cells kind of reach the maximal speed they can. And we are trying to boost the response in different ways. So I have new collaborations that I'm starting well recently, and that I will explore in the future. And then we have found some other ways to boost the response. It's not necessarily the two different stimuli, but maybe boosting some of the particular signaling. And I hope in the near future to tell you some stories about that. But it's a great question, yeah.
All right. Then move on to the next question from Guy. Sorry. So DAMPs constitute a character of molecules, not only ATP. So, for example, what is contribution of other DAMPs beside ATP in dendritic migration? And the second one, what is the contribution of specific ATP in the complete DAMP cocktail for the dendritic migration?
Oh, that is a great question, Miguel. Actually, so we took it isolated, right? So then we just used ATP by itself. So then there was not mix of danger-associated molecular patterns. People do this to induce the activation of dendritic cells. So then you can actually, people is mixing matched cells. So then essentially people match cells and then they deliver this to the cells. And that contains the full mix that here is talking about. So then it contains chromatin, HMG proteins, and also ATP and other molecules. We did only ATP, but we have other molecules too that we haven't published yet, and there are other DAMPs, and they also increase the speed of migration, but the response is different. So the time they take to induce the increase in the speed is not the same. So there is a different kinetic, and the combination of them is totally unexplored for the moment. So then, yeah, what is the specific combination of ATP in the mix? Well, we tried ATP by itself, and then that's what we have. And in the cocktail, we haven't tried yet.
Okay. All right, following question, Mohammed. So any idea why BAFTA is not inhibiting calcium spike in MYO1E SI condition?
So we think, so the one cell I show, it still has some oscillations. The compilation of many cells, then it shows that a significant decrease afterwards, and then they actually stop. But we think that maybe this myosin 1E knockdown cells, they use the lysosome or any other reservoir and really keep it. I don't know how they could chattel, but it seems that they do anyways. I haven't tried for example, mixing BAFTA and EGTA or EGT alone. So this is one of the last things we want to do just before submission. But yeah, it is a key point to respond yet. Yeah.
All right. Martin again. So what is the size of the local calcium signaling that you showed? How frequently do you see them?
Well, that was an isolated observation in that particular case. I haven't quantified it yet, so that I couldn't. It's a great question. I don't know how often they are, so then that is something really cool that I would like to explore. The size was should be around a micron, so half a micron I would say half a micron. It is quite big to be a nanodomain like the ones described by the Guz's lab, but then and others. But then I don't know what it is actually. That's why I would like to study. I haven't seen the frequency, but it's not only that cell that has it, but I haven't quantified it. But I have seen in several cells, and what intrigues me is that sometimes it occurs between waves, which is more interesting, right? So then the wave stops, and then the local increase occurs.
All right. So the following question: which mode of motility are dendritic cells using in the microchannels? It did look like methane chemicals and laminopodia, but what do you think about mode of the motility?
Yeah, right. So then the dendritic cells, they use an amoeboid migration. They don't use mesenchymal migration actually. So yeah, that is a great point. So a mesenchymal migration used by, for example, by fibroblasts or endothelial cells is not the case for dendritic cells. They use amoeboid migration, and then it's not non-degradatory migration. So they don't degrade the microenvironment with ectoenzymes, but rather they deform. So then, yeah, there is no a real mesenchymal lamellibode because they migrate in an amoeboid manner, and they sort of keep the shape. They are more compacted. The cells with a long lamellibode. But there are conditions when we can change the adhesion of the surface, and then we can make them migrate in a more mesenchymal way. But what I show in the microchannels is amoeboid migration.
Okay. Yes. The next one. How fast do you record moving cells in the microchannels? Could it be that the layer accumulation is just an effect of the capturing of the image, then adding them together?
So that is an important point. So then it could be an artifact if the rate of acquisition was slower than the moving itself. But in this case was spinning this analysis. So then spinning this microscopy, you just take a snapshot of the full cell at the time. So there is no scanning like in the confocal where you will maybe add up because of the moving of the cell, the motion of the cell. In this case was a spinning microscopy. We can go as fast as up to the second scale, and then we observe the same. Also, we did it in fixed samples, knowing where the cells are coming from and where they're going. So because we can put the cells in the one well and look how they are facing to the other side, and then we saw the same. So yeah.
Okay, I think following up, can you see a calcium gradient in migrating cells, high in the rear end and low in the front advancing edges?
That is a good question, but we haven't analyzed it. So this is a classic, what is called the paradox of the calcium paradox in migrating cells, that is higher at the rear and lower at the back, at the front, where there might be some local spikes. That's what we aimed to see, but actually we haven't seen that. In the microchannels it was very hard, and then to our suffering, UV illumination would absolutely stop the migration of the cells. So we cannot use the classic FUTA2, for example, and others. So we try to do ratiometric analysis with other probes, for example, Oregon Green and FUTA Red or stuff like this, but we couldn't really see a gradient in the channels. Maybe that is different when there is a chemokine, and then because in the microchannels is spontaneous migration, so there is no chemokine at all. Maybe when there is a chemokine, now the cells would actually have, maybe, we don't know yet, they could have this gradient, the intracellular gradient.
Okay, so why would the dendritic cells express MyoE in the first place? In other words, what would be the advantage of these cells to have a MyoE at all? What would be
that is in B cell. So in B cell, yeah, that is a good question, because why there would be, what would be the use of a negative signal in the to prevent the response? Well, the same that the checkpoints. So then immune cells, and then in the immune response, always have an inhibition of the response to avoid the response to get overactivated. So we think that there is a different kinetic, and could be a sort of tug war between myosin 1C and myosin 1E, and if there is some imbalance, then the response will be higher or lower. But probably just because of the kinetic of the response, myosin 1E is winning over the time, so in this way prevents the overactivation of the cells. So then that's what we actually think. Initially, anyway, we had to say that this was completely unexpected, and we initially thought only they will play a role in the cytoskeleton. We never thought of it to play a role in the calcium signaling directly by modulating PLC gamma 2 activity. But one function will be the cytoskeleton role for the actin, and then we will have the other one for the signaling with the PLC gamma 2 to prevent the overactivation of the cell. That's what we think.
Okay. So the last one, do you expect to find myo1E and myo1C at the same position as actin?
Yes.
Okay.
So yes, the myosin 1C and E, they arrive at the synapse of the B cell. They arrive at the same time, more or less, and they are localized right there at the synapse. But then we haven't done like super resolution to see whether the myosin 1E maybe is more of the borders of the synapse and the myosin 1C in the center. Dendritic cells do express myosin 1E and C too, but they express more homolog. So for example, myosin 1E, which is the long tail, dendritic cells express high levels of myosin 1F. That is also a long tail myosin. And the myosin 1C is not so expressed, but they express more of myosin 1G, which is also a short tail. We haven't analyzed that, and then that is most of the work of Ana Maria's lab. I think that maybe she will continue doing so. I don't know.
Okay, I think this is the last questions, and I think thank you very much to everybody joining this webinar, and thank you very much, Pablo, for nice presentation and nice discussion to everybody.
Thanks for watching, and thanks for the people that stay here. Yeah, if someone else has some more questions, I will be keen keen to discuss now or in the Slack channel of the society.
Thanks, Pablo. Thanks, Sachi. That was really interesting and lovely slides. Really nice slides. Really well presented. I have a kind of an informal question. When you showed your TRPML1 knockout data, it looked like the TRPML1 knockout cells were slower, and I think you showed significance on that graph. They were slower than the control cells. So did they just have a general defect in movement that they couldn't then recover with stimulation? Because it would have been nice if they'd been exactly the same to start with and then not have the agonist response rather than being a bit lower already.
That's correct. We think that there is already a defect, as you said, so then that it is the case.
Yeah.
But and they still kind of preserve the ability to speed up.
Yes,
that is true. So with the other Pablo, we had this project that we couldn't finish. We had too many things to do, but the idea was to see which channels, and that's what I'm analyzing now, and I have some data for that.